Great, very good. Actually, we are in this chemical engineering and materials engineering department, and we are doing very much soft side of what we do as a department. So I'm originally a material engineer, a polymer engineer. So polymer is a long molecule, like a large macromolecule. So I got my degrees from polymer engineering and macromolecular science and engineering. And that was my original background. And then after I got my PhD degree, actually during my thesis research for my PhD, I got into got interested in this biomedical field. So how can I use these biomaterials that is made of this long molecule like polymers? How can we utilize those molecules for unique applications in biomedical side? So I studied afterwards, after I got my PhD degree, I studied with a professor at the medical school, immunology department, to study about our immune system, especially the adaptive immune system. It is called T cells. So that's why our lab now, in my lab, we are studying essentially the two arms of research, basically. One is the polymeric biomaterials, and the other arm is immunology. And then we are combining these two fields together, okay? So okay. So I prepared this short slide to give you some background knowledge. I'll just, not too much in depth, but Terry, you heard about this COVID-19 vaccine. These days, everybody is worried about infection and how can we protect ourselves. And this may give you some background knowledge to understand better of the whole news articles. So our body is exposed to this variety of different pathogens all the time. And then when these pathogens or like bad guys, the viruses and bacteria infect us, we have in our body, there are a set of cell types that are always surveying around, whether we are infected or not. And then these guys provide so-called cellular immunity. So it is a immunity provided by these innate cells. So innate means no matter what that infection is, these cells are providing some protection. So how do they protect us? They detect these bad guys, engulf it, take up, and then digest it in the non-functional forms. Okay, but that's not it. So sometimes, if the infections are too much, or if they are not recognized by these these cellular immunity components, then they just kind of break that line of protection and then just start to infect other organs and cells. So but then these small number of innate immunity, those cells that we have in our body, kind of, as I said, they digest. They make these pieces of bad virus and bacteria presented on their surface. Okay, presented on the cellular membrane. Then the next series of cells start to come in. So those are called T cells. Okay, and these T cells have special receptor. Okay, receptor is a molecules that can recognize this special molecules on the surface of this antigen presenting cell. So those cellular immunity component is called antigen presenting cell. They are presenting the antigen, and then the T cells are recognizing them and then get activated. And once they get activated, now it's getting more complicated, right? So once they are activated, they can actually help, okay, help other special type of immune cells called B cells, okay, B cells. So it's a T cell from the thymus. B cells is from originally from bursa, but you know that's why it is called B cell. But B cells get the help from this helper T cell, and then it expresses some specialized molecule called immunoglobulin or antibody. You probably heard about antibody, right? So these antibodies are expressed on the surface of, originally expressed on the surface of these B cells, okay? And these B cells, when they get this help, you know, cell to cell communication, and then get the help from the helper T cells, then this B cells basically change its form. It's called differentiation. It changes form to, you know, some functional form of cells, so-called memory B cell or plasma cell. And these plasma cells are responsible for making this antibody. So you probably heard about this antibody titer against to this virus, COVID-19. That's the reaction that we just talked about, right? It is coming from the reaction, the collaboration between the APC and B cell, T cell, and then eventually this plasma cell secretes out this antibody. Okay. Is there any questions so far? Okay. So that interaction that we just talked about is happening in our body. Where does it happen? We have over our, all around our body, there are lymph node, okay, lymph node. And actually that happens in the spleen as well. But bottom line is this T cell, B cell communication, and then the B cells changing it to this functional form that is happening inside of this lymph node. So that's why when you guys get cold or, you know, like some infection, you have some like an enlarged mass you can just detect somewhere around your neck. That's the lymph node becomes bigger, okay? So your B cells and T cells are starting to work, okay? So in our lab, we kind of simplify this interaction between the B cells and T cells, and then we recognize, aha, there are a very special pair of molecules called CD40 on the B cell, and then CD40 ligand on the T cell. These bindings makes the B cells to do what we were talking about, right? Activate it and changing the form, you know, secreting the antibody and all that, right? So in our lab, okay, so Michael now during the summer, he started to discover some new ligands that can bind to these CD40 molecules on the B cell, okay? So so again, we are studying this simplified model of B cell T cell interaction. We are developing something that can replace the CD40 ligand on the T cell side. Okay. So Michael.
So CD40 specifically is a protein. So it's a polypeptide, series of peptide molecules, amino acids in a certain sequence, but then fold together into a certain shape. Mm-hmm. And you can see that shape here on the screen. And we use some molecular modeling software to produce that. And that is what the protein actually looks like. If you zoomed in on it really, really far, that's what the molecule would look like under a microscope. Mm-hmm. And that is our target. We're trying to find other proteins that can bind specifically to CD40. We're trying to find which ones can and which ones can most effectively. Mm-hmm.
Michael, what do the arrows represent? Or are you going to get into that? The arrows in that shape.
Anything about this arrow?
The arrows just show the direction in sequence. So there's a start to the sequence, there's a first amino acid, and then there's a final one. And that's, the arrows are just showing the direction sequence-wise. So it lets you identify where it folds and how it folds based on that.
Yes, thank you, Michael. So the amino, so protein is just, it's actually polymer. It's a long chain molecule, if you really imagine. So it is an amino acid after amino acid. It is connected. And then there is always start to the end. But this proteins or polypeptide never exist as a line. It folds into this three-dimensional shape. And then as Michael just said, this arrow is added as a scheme to show that that's the direction of the amino acid, how it is connected from the start to the end. And
so here we have a larger interface, and it looks kind of nuts, but this is the interface between the CD40 ligand on the helper T cell and CD40 itself on the B cell. So right here is CD40L, and it's bound to CD40 there and there. And there's another molecule of CD40L bound to CD40 right there. And that is the goal, is to find something that mimics CD40L in such a way that it will bind to CD40 and hopefully activate a B cell.
So these amino acid has so-called side chain. So when these polypeptides are near each other, this chemical, the bond, so-called, some interaction, you probably heard about this, van der Waals force, hydrogen bonding, and those interaction makes them to interact each other. And these two, CD40 and CD40 ligands, they are designed, or they actually binds to each other in a certain form, certain conformation. And Michael picked this, simulated image to show you how the CD40 and CD40 ligand interact, right? Oh, I'm sorry. Yes. All right, sorry. Okay, so what Michael is doing this summer is actually using the general technology that these three scientists got awarded for in 2019 for the Nobel Prize. So these three scientists got a Nobel Prize for basically proposing and showing the proof of concept for the last 20-30 decades. So what they did is actually they engineered some genetic information inside of these bacteria, bacteriophage. So these are the virus for the bacteria. It is not important. So this is basically some particle shape of virus, and they actually change the genetic information inside of this virus so that they can express a bunch of different types of ligands or polypeptide or short peptide on the surface of this virus. So, and then what they did is actually they okay, so let me I will just stop there and then let Michael explain more about it. So anyway, so that's called biopanning. So as I said, so what the first stage of this experiment that Michael didn't do, but what we are using here is, again, So there is a DNA, circular DNA, that is encapsulated this viral particle. This is a virus. And then we, as a chemical engineer, we can actually modify this gene that encodes all these proteins that makes the outer side of the virus. And then put something randomly mutated gene into this circular DNA. Then what it does is actually it expresses the corresponding piece of peptide at the surface of this virus. Okay, and by changing this sequence to billions, millions, and billions of different kinds, okay, randomly mutate this DNA, then what do you expect? Yes, we have millions and billions different kinds of pieces of peptide that is expressed on one side of this viral particle. Okay, so we call that as a library of this peptide. Okay, and the library is expressed on bacteriophage, so we call that as a phage library or simply peptide library. Okay, so we are using that peptide library, and then this is the scheme that Michael has been doing over the summer. So Michael, can you?
So in our specific case, if you look at the target coated surface down here, we're using magnetic beads to do that. And it has a certain protein on it called protein AG, which binds to a certain protein that's been attached to the end of our target molecule, allowing us to essentially magnetize the molecule we're trying to bind to so that we can easily separate the phage.
Mm-hmm.
So we're going to take that bead and add that phage library. So it's going to be a bunch of random peptides on the ends of viruses. They're all going to be reacted together, spun around, mixed together, and the ones that bind to the target will stay on the target. The ones that don't will keep floating around. So then we wash away all the phage that doesn't bind, because that doesn't matter to us. It's not what we're looking for. Then we have all the phage that we have bound, and we take an acid to break that bond between the phage and the target. We remove the phage and the acid, neutralize it, and then do something called amplification. So the cool thing about viruses is they like to reproduce. They want to make more copies of themselves, and they do that by infecting bacteria, injecting their own DNA, and sort of tricking the bacteria into producing a bunch of the virus. And so if we grow E. coli and add our phage that we have that's bound to our target, we can get a bunch more phage by having it naturally just amplify itself and increase greatly in number, about 10,000 fold. And then we repeat the process with all the phage we got from the first round. And we do the same thing. We bind it, we wash it, we break the bond, we amplify it again, and we do this maybe 1 or 2 more times, and then isolate the DNA from the phage and sequence that DNA to figure out what peptide was on there.
Mm-hmm.
And that's the end goal.
Yes, that was the end goal. So, okay, sorry, just bounce back. So this is a starting point. So we started with billions of different kinds of these molecules on one side of the viral particle. And then once we incubate with the target, this non-specific guys, which doesn't have any affinity towards the target, will be washed off. Only some viral particles that has the affinity, like this key and lock type of interaction, will bind. And then as as Michael was saying, we elute them and then amplify them and then do this process one more time. So then out of this, amplified phage, more higher binder, tighter binder will bind, and then the less of a binder will be washed off. So after these 3 rounds, we will potentially have some particles that has very, very high affinity ligands attached to the target. And then, as Michael also said, by looking at the DNA sequence, okay, so the DNA, the beauty of this technology is this: the DNA is essentially dictating the peptide, right? So now we know what DNA, by looking at the DNA that is isolated from this virus, we can actually know exactly what type of peptide will give us this high binding. Okay. All right, so Michael, so this is
an example of our
work.
plated bacteria. So one cool thing about bacteria is if you give them food, essentially molecules that they can eat and turn into energy, you can grow them on a plate. And our bacteria has been modified so that when it is infected by phage, it will make a blue plaque. So all of the black Sharpie dots there are us counting our blue plaques to try and figure out how much phage we have. That allows us to calculate something called a plaque forming unit. And that gives us an estimation of how much phage we have and how much we got on return from how much we started with, because we counted the plaque forming units for that as well.
Correct. So Michael, basically, basically started with certain number of viral particle for the panning, and then after the incubation and washing and elution, he gets a certain number of viral particle as well. So what he was saying is basically by plating the infected bacterial cells on the plate and then counting the number of plaques, we can back calculate the amount of starting viral particles by using this method. So, we started with a bunch of pipetting and still doing a lot of pipetting. So, but it has been working pretty nicely. So, what do you do after that amplification?
So our final step is to purify the viral DNA. So in order to do that, we centrifuge, so we spin the bacteria around at really high speeds, which breaks the cell wall of the bacteria and causes the cells themselves to go to the bottom of a tube where we can remove everything else which has our phage in it. And then we use sodium iodide to break the phage open, exposing the DNA. And then we use ethanol to precipitate the DNA out of solution and into a little pellet. It's this off-white kind of beige looking pellet at the bottom of the tube. And then we suspend it in water and mix it around a little bit. The DNA gets all spread out evenly through it. And then we send that to UAB, who runs something called Sanger sequencing, which is a technique for figuring out what the DNA sequence of a certain thing is. And we will receive sequences from them. And you can receive good sequences like the one at the bottom, or like we did at some point, bad sequences here at the top. And this was the big problem we've encountered so far this summer, is these bad sequences. So if you look specifically right here, the results of a Sanger sequencing will give you a graph of the concentration of a certain base, a certain molecule in DNA that makes up the DNA. And there'll be different colored peaks. So here with this N, that means we couldn't figure out what base it was. And you can see that there's a overlap between the blue peak and red peak here.
Mhm.
And that makes it impossible to tell whether it was an A or a C. In this sample down here, which is a lot better, you could see these clearly defined peaks where there's no overlap, there's no other thing rising up. It's just a peak of one color and then a line at the bottom. And that's what we're looking for. That's what we've been getting more of lately. And
Yes.
that's
So
been the hard part, is figuring out what's gone wrong and going back in and repeating the process, re-amplifying our phage, doing it exactly correctly in order to get DNA samples that
mhm.
are clearly readable and allow us to have no ambiguity in what our peptide actually is.
Yes, yes, yes. That Michael really explained it nicely. So, so this Sanger sequencing that we are using, I mean, at the UAB, the center is using, is essentially using some fluorescence tag that is given to this ATGC pair. So that's the nucleotide that makes the DNA, the long chain DNA. And if you have one peak, so here, as you can see, the C is tagged to blue, A is tagged to red, T is tagged to green, G tagged as yellow. Okay, so if one particular end or sequence is either showing one of these four peaks, then we can definitely tell that, oh, okay, so this sequence is made of this sequence of ATGC, mixture. But as Michael was saying, if one position has more than two peaks, okay, significant peaks, we cannot necessarily determine what exactly that position is composed of, right? So then there could be a bunch of different reasons. Michael spent a good 2 weeks or so to figure out what was wrong with this his sample. And then, but he really did a good job rigorously studying the protocol, going back and tackle this issue. And then at the end of the day, he figured out basically the steps where he actually lysed the virus was not properly done, right? And then he got later on, after he figured that out, he got much better quality DNA. So then now we have always get a very nice sequencing out of this sample. So yes, we are actually going, still doing this, right? Second round, third round, we're having a lot of fun. I just wanted to share this video. So, okay, so what can be done after we figure out this peptide ligands? So this is an example video that we published in our lab sometime late 2020 or early this year. So these are the video of this micro beads. It is artificial beads, and then these big round shape is immune cell. In this case, it's a T cell. So we did coat these beads with this peptide ligands that we found in the same technique that has an affinity towards the molecules on the T cell. And then I'll just play one more time. So these T cells are actively looking for and interact with these beads. And look at this, like the cells are touching the beads, and then it basically, because of this ligands coded on the beads, it interacts, and then it basically get the signal, right? It is touching, like almost like a, touching and sensing. And then it is showing the typical behavior when these T cells are fully activated. It becomes round shape and then resting. So it was an interesting result. And then with the similar stuff, when we have this sequence of peptide, then we can synthesize this peptide, and then combining with this biomaterials platform, such as this microbeads, and then, or nanobeads, nano-sized particle, and then we can use that as an activator of B cells, so that our B cells can do better job in making antibodies. Or actually, we can do this culture of B cells outside of the body, so that we can make these B cells as a therapeutics. So some patient, they do not do this immune reaction inside of the body, but we can take out the blood samples from the patient, and then do this culture of a B cell and put them back into the patient. Or we can actually, so when the B cells are activated in a wrong way, then it can attack our cells, some organs, okay? The antibody that is secreted by this set of B cells can attack our own body. So in that case, our peptide-based therapeutics can prevent that from happening. So then in that case, this peptide can be used for therapeutics for autoimmune diseases. Okay. So, okay, that was it. Yeah. So, yes, so this is our website. If you are still interested in what we are doing, you can visit us on the website. With that, thank you for your attention. I'll just ask you if you have any questions. We'll be happy to answer.
I have a question.
Sure.
Okay, so you know how you had the phases of the the ADC and like how it mutates. Does it just keep does it just keep repeating itself over and over again, like after, or does it get farther into the mutation?
So you mean the cycles of biopanning?
Yes.
So in our case, we start with a bunch of random mutations, and then we try our best to not allow it to mutate. So you're probably talking about what Smith did in his paper, where he randomly mutated during the process. And that's something that we're choosing not to do, because it's our goal to find peptides that are the most specific to our target. Mm-hmm. His was more of a proof of concept, where he was trying to prove that you can bind a phage to a specific molecule and then keep it, discard the rest, and continue to bind them. And he added mutations to prove that you could continue to discard the ones that don't work.
Yes. So Michael's experiment project started with this bunch of different phage that has already the mutated form of peptide on the surface. So we are not inducing any more mutation. If that's your question, does that answer?
Yes, sir.
Very cool. Excellent. Any other questions?
Yeah, I have a question. So you mentioned that the virus itself doesn't really matter. This works with any virus body. But do you see, or is there any potential for working better with any particular virus?
So we do use a specific virus called M13 bacteriophage because it proliferates quickly and we have its genome very well sequenced to the point where we've identified the exact portion of its genetic code that we can modify to change that protein. And we also have it well sequenced to the point where we know the genetic code right before the random mutation that allows us to go in and very specifically sequence that DNA and find the exact portion we need without going through 6,500 base pairs of DNA, which is quite a lot. And so we're just using that specific kind because it's useful for what we're doing and we understand it very well.
Yes, thank you, Michael. That's a great answer. Actually, so when I say it is not important, that's completely wrong. So all the details are important in science. So actually, the viral display, in this case, the viral particles were chosen for the specific reason that this virus can infect the bacteria, so that the bacterial cells can grow and amplify, make more of a phage very soon, very easily. So bacterial cells grow so fast. So that's a big reason that why people use this virus that infects bacterial cell, for that reason. But sometimes if you want to make this library for a bigger form, bigger protein that mimics the protein that is expressed by these mammals like a human, then actually people use completely different platform, the same concept, but they actually express this library on the surface of yeast or even bacterial cell itself. So bacterial cell yeast cells can become the platform that is replacing the virus in this case.
Okay, that makes sense. Yeah, thank you.
So as a specific example of what Dr. Rowe was talking about, the amplification step, where we end up with about 10,000 times more phage than we started with, takes 5 hours. It's less than one workday to amplify our phage and get 10,000 times more of it.
Yes, that's
how quickly our bacteria grows and how quickly our virus is able to infect it and continue to grow.
Yes, that's a huge advantage.
That is really cool. Yeah, Tyree, this project basically takes 10 weeks over the summer. So especially in projects like this, where there's lots of time consuming, waiting for things to grow, and then these really complicated processes, which if something goes a little bit wrong, you have to backtrack and figure out what that was, like Michael did. So I'm sure he's been super busy, and this program ends next week. And so already I'm saying, okay, now you got to make your poster, Michael. And he's probably, about ready to pull his hair out. But it's really, fast paced and fun kind of stuff. But definitely being a researcher, you have to be somebody who, can deal with adversity, and when something goes wrong, try to figure out what happened and backtrack. So it sounds like Michael's done a really good job and shown really good researching skills.
Yes, he did.
Any other questions, Tyree?
Yes. My other question is, how the limit of the amplifying of the bacteria, can it go over 10,000?
Yes, it can. So the limit for our bacteria is we can grow it for about 16 hours before it stops growing quickly. So it grows on something called a logarithmic scale. So it'll start growing slowly and then it will grow very quickly and then peter off again. So 16 hours is about the point where it'll stop growing quickly. And we could get more than a 10,000-fold return if we amplified it with more phage, more bacteria, or more food for the bacteria. So we use something called LB media or LB broth, and it contains something called bactotryptone, which the bacteria use essentially the same way that we would eat a meal. The bacteria will eat it and convert it into energy to grow. And if we use more of that, we'll get more bacteria. If we grow it for longer, we'll get more bacteria. And if we start with more phage, we'll get more phage at the end. So it's entirely possible that we do that, but we've found it the most efficient to do it for about 5 hours and pull it there, because anything else requires you to grow the bacteria for far longer. And it would turn a 1-day process into a 2 or 3 day process, which would slow us down greatly. That's a really good question.
And I have one last question. And you know how you said that after, like, you send the bacteria to UAB and then they test it? What do they do after they test it? Like, where do they store it at, or where do they put it at?
So when we send our DNA to UAB, they will store it in a negative 20°C freezer. And when they're done sequencing our DNA, they will have completed a reaction. So Sanger sequencing involves something called a PCR reaction. So you run a chemical reaction on the DNA, and afterwards they will send us the sequence in a certain file. They'll email it to us, or they'll post it on their website for us. And then they will most likely dispose of the reacted DNA while keeping the non-reacted DNA in the freezer in case we ask them to run a certain sample again, because we want to check the purity or the quality of the DNA or the sequence itself. So they'll get rid of the stuff that's been reacted because they can't go back in and react it again, essentially. Does that kind of answer your question or make sense?
Yes, sir.
Excellent.
All right. Oh, I think someone popped in, must be for the next one. So yeah, thank you very much for your time, Dr. Roll, Michael. That was really interesting. Tyree, thanks for coming again. It was really nice. I've seen you at, I think this is probably the 4th or 5th one I've seen you at. So I hope you've enjoyed all of these. But anyway, yeah, when you come to UAH, these are projects that you can get involved in if you're interested. And so I encourage you to definitely check it out. Check out their website. You can see their work continue. But otherwise, yeah, I just want to say thanks again. And everybody have a great, let's see, it's not Friday yet, but almost Friday. So weekend's coming up. Have a great weekend. And we'll see you next time.
Thank you very much.
Thank you.
Dr. Cook, Tyree.
Thank you.
Bye.
See you.